My name is Susana Alcantara, and this webinar we will discuss the basics of live cell analysis applied specifically to the neuroscience research field. I will illustrate different concepts using relevant examples that you can hopefully find useful for your own research. I really hope that you find the next 25 minutes interesting and useful. The last few decades have seen a large number of failed clinical trials in the neuroscience field. Therefore, whilst this area has great potential, it is now regarded as higher risk. The main challenges I believe that neuroscience research encountered are the extraordinary complexity of the human brain and its plasticity, making it difficult to model in the lab. The difficulty of accessing human tissue and the ethics behind it. The questionable translational value of animal research models. As you know, we are still identifying neurons in humans which are not being previously seen in rodent brains. And finally, the extreme sensitivity that characterizes neuronal cultures. Now, having said that, the field of neuroscience has also witnessed remarkable developments in more recent times, and there is an increased investment towards this research area once again. An important advance, for example, is the development of human hiPSCs, which presents a huge potential to generate fully humanized, patient-specific advanced cell models. When coupled to more advanced techniques, one can hope it may yield better translation to the clinic. There are currently a great number of possible cell-based strategies to model a particular disease, from immortalized cell lines to more advanced cell types, such as patient-derived iPSCs, and from 2D up to organelles. While cell lines and 2D cultures still provide valuable basic information, the increased complexity of advanced cell systems, I believe, can potentially lead to an increased translation to the clinic. These advanced cell systems are composed of precious cells due to limited supply or higher costs. They provide more dynamic systems with increased plasticity, which, if studied using the right technology and appropriate analytical methods, can be information-rich, therefore translating into more relevant disease information. Now, for this reason, we need to develop cell-sparing workflows which allow us for temporal monitoring and relevant, non-perturbing analytical methods. The common technologies currently used in the neuroscience field are capable of measuring various aspects of cell health, morphology, and function, but we need to take into consideration, firstly, that multiple pieces of equipment are necessary to answer the above questions. And secondly, that to move science forward faster, one needs approaches that are information-rich and can track dynamic biological processes as they unfold. One of the aims of this webinar is to show you how well-suited live cell analysis is in the development and validation of advanced cell models. So firstly, what is live cell analysis? It is a continuous or semi-continuous cellular measurement, and it needs to provide relevant information in real time. It certainly needs to be obtained from living cells, and the data acquisition should be non-perturbant to these cells. A direct consequence of this is that these measurements are non-exhaustive, meaning that once the assay is finished, these cultures can still be used for secondary downstream endpoint analysis if necessary. Now, lastly, it needs to be applicable across workflows, so that ideally a single instrument is flexible enough to answer multiple questions on morphology, health, function, and dynamism, and can provide information during all research phases, not only the assay phase. We have here a comparison of traditional endpoint workflow with that of live cell analysis. The only analytical data generated by the endpoint approach is a single and arbitrary time point obtained during the assay phase. In contrast, live cell analysis offers continued information during culture, manipulation, and finally the kinetic assay. So how do we go about creating a technology capable of truly performing live cell analysis on those terms described before? Well, the data I'm about to show you has been acquired using the IncuCyte live cell analysis system, which is a platform, an integrated solution of hardware, software, reagents, and applications, all designed to work together. The hardware is a microscope optimized to permanently reside in the standard lab incubator. It takes images from cells cultured in different types of vessels, like flasks, plates, petri dishes, etc. The software stores and analyzes the images to extract phenotypic data in real time. And all this comes with reagents and applications which have been optimized by the R&D teams to which I belong for the use in this system. In this webinar, I will cover 4 major research areas, and I will provide you with relevant examples which will show you how one can automatically quantify the cell health of your cultures, the neurodynamics and neuronal activity, as well as the neuroimmune function. All this using live cell, non-perturbing, real-time analysis in the way described before. Let's start introducing the first area, neuronal cell health. In this first example, I will show you the use of Annexin V to understand cell death of your cultures. This reagent, as you know, binds to exposed phosphatidylserine, which is one of the hallmarks of cell death. It releases a fluorescent signal which increases with death. So briefly, cells are seeded and treated with drugs diluted in media supplemented with an Annexin V reagent. Images are taken for the necessary amount of time and automatically analyzed. You can see the onset of death in the images below. This experiment has been performed in collaboration with CDI. Agglutaneurons in monoculture were plated in 96 well plates and cultured for 10 days. After this time, they were treated with increasing concentrations of glutamate alone or in combination with the NMDA antagonist MK801, all this in the presence of an Annexin V orange reagent. Cell death was recorded for a further 72 hours. The greatest assay window was reached within the first 24 hours post-treatment, time at which the CRC graph was derived. Now, a different visualization of this experiment is represented with the plate view on the right-hand side, in which each square represents a well, and within it, you can see the temporal response of an Annexin V orange intensity signal. The next research area I wanted to discuss today is the ability to quantitatively measure neurodynamics in monoculture in a non-perturbing and non-exhaustive way. We call this the NeuroTrack application. Neurons are plated, treated if necessary, imaged, and neurons and cell bodies are analyzed in real time and automatically segmented. You can see the masks in yellow and blue respectively. This method obviates the need to fix and stain in order to obtain neurodot growth readings, saving time, decreasing experimental artifacts, and enabling the visualization of plasticity. These assays perform label-free, and the sensitivity is such that one can obtain differential profiles in the development of networks from, for instance, different primary brain regions. In the example shown on the right-hand side of your screen, one can compare the speed of development of cortical, striatal, hippocampal, and midbrain neuronal networks. The type of data that we may be able to study using the NeuroTrack application is, for example, the non-perturbant differentiation of cell lines or neuroprogenitor cells, as in this example on the left of your screen, in which the video and its temporal analysis represents the process of differentiation of human iPSC-derived neurons obtained via collaboration with Essen BioScience. This can be applied, for example, for QC in proposes or workflow optimization. Another application that may be compound profiling, for example, during drug discovery projects. Again, I would like to stress that those cells have not been perturbed in any way, so that by the end of this experiment, they can still be used for downstream secondary endpoint measurements if necessary. Now, this application is amenable to monoculture only. When a base of cells exists, as with the study of neurodynamics in co-cultures, the introduction of a fluorescent marker is required. Assays contrast images alone are not able to discriminate the neuronal projections from the background of support cells, as you can imagine. For this reason, we have developed the Neurolite orange reagent, which is a lentivirus that encodes a fluorescent protein. This protein is driven off a synapsin promoter to strengthen neuronal expression and minimize non-neuronal crossover. With this reagent, we can enable labeling that is neuronally specific, long-lasting, and non-detrimental to cell health. Here we show a kinetic adaptation of the well-known model, a XOHDA model of neurodegeneration in Parkinson's disease. Primary cortical neurons and astrocytes were co-cultured and infected with Neurolite. We then waited for 14 days until we observed that neurodevelopment was established. Then the co-culture was treated with various concentrations of the neurotoxin 6-OHDA, readily used to kill neurons in this model. Neuronal growth measurements continued for 10 more days. And now we're moving on to the third research area, the neuronal activity. Now, during this webinar, I especially wanted to touch on this new application we've just launched, which I believe is of great value and has the potential to enlighten us with important insights on the biology of our neuronal cultures. I will try to convince you about this within the next few slides. As I mentioned before, the IncuCyte is an integrated solution composed of reagents, optimized protocols, software, and hardware, and this application I'm about to introduce you represents an example of this. The neuronal activity application uses a new reagent, the NeuroBus orange, which is a genetically encoded calcium indicator. In the presence of calcium, calmodulin undergoes a conformational change and forms a complex which then emits light. This reagent enables the functional study of actively firing neurons by reporting calcium oscillations that can be seen and measured in the orange channel. The protocol is a simple lentiviral infection of your co-cultures with NeuroBus. If necessary, one can let the network stabilize before adding the required treatments. Imaging of these cultures can start at any stage during this process. In order to see and measure the calcium oscillations, we've developed a new acquisition mode. what we call as the STARE mode, which takes images at high frequency during up to 3 minutes. The video here shows you the 3-minute epoch of highly synchronized I-gluta cells from CDI co-culture with primary astrocytes. The software automatically segments these high-frequency images so one gets the information in real time. Just to add, the NeuroBars Orange is compatible with a variety of neuronal cell types, it's non-perturbant, and it maintains expression over weeks or months. In summary, this application enables the long-term monitoring of neuronal excitability and network activity in co-cultures. We see here the type of data obtained with this module. The image is a collage of single wells seen in phase to qualitatively assess cell health. Orange, which gives us information on areas with calcium influx, and its segmentation, indicating the presence of active bursting nodes. The IncuCyte software automatically calculates an activity plot summarizing the change in activity of each pixel during the 3-minute epoch. Each individual calcium oscillation from each active node within the 4x field of view is recorded and represented in a superimposed calcium trace, as the one you can see above, where each line represents a node. For a seed of 25,000 cells per well, for example, we normally achieve around 1,000 active nodes. Plating density can be optimized by the researcher for their particular experimental needs. Now, if we repeat the measurement every 24 hours, and of course this can also be modified based on research needs, we can follow the development of network, its connectivity and stability as seen in the middle bottom figure, enabling the study of plasticity, for example. The 96 well plate views on the right represent the calcium summary trace in each well at a single time on the top, or the concatenation of metrics in the bottom, to provide a long-term picture of activity. This enables the study of, for example, slow modulation during long-term neurodegeneration. Before I continue with a translational example of this technique, I would like to show you some validation we are all used to seeing. In this slide, we will show you how one can modulate neuronal firing activity by using drugs such as the stimular GABA antagonist, picrotoxin, or the potent neurotoxin, TTX, and how this can be validated using the integrated movie viewing tool. Looking at the control well, we can see sustained neuronal activity. Picrotoxin displays an increased activity and connectivity. This is represented by the calcium traces below each movie and described by the mean correlation metric. Neuronal activity is abolished by TTX, as expected. While both drugs showed a dramatic effect in neuronal activity, phase images don't display any apparent changes in the morphology of the co-cultures. To obtain an increased biological insight into your research systems, one can look at the effects that these treatments have in the bursting profile. We can see where we can study the number of active nodes, mean correlation, burst rate, or intensity as shown within the bar graphs below. On the left-hand side, we can see the profile of the baseline activity of our untreated culture. We can then compare this baseline with the effect of picrotoxin treatment in the middle to understand how these bars have been modified. With a similar number of active nodes as control wells, the bars are more synchronized, more frequent, and of higher intensity when compared to vehicle, as expected from this stimulant. Thus, lines represent the control values for an easier comparison. In contrast, TTX treatment almost entirely stops activity in the co-culture. This example shows the power that this technology and its derived metrics can potentially have to distinguish among drug treatments for the drug discovery field, for example, or genetically modified cells for disease modeling. Another interesting example I wanted to share with you is the capability of using neuronal activity to compare the functional phenotype of different neuronal types, and so being able to characterize them. We have here iPSC-derived GABAergic and glutamatergic neurons from CDI. One can characterize them using the previously described metrics. The data presented here allow us to easily see that non-excitatory GABAergic neurons on the left exhibited spontaneous activity, but only the excitatory glutamatergic neurons achieve coordinated network activity. One can imagine that this type of sensitive characterization may enable the investigation of disease versus not diseased neurons, neurons from different brain regions, or even neurons primed with brain extracts from both healthy and diseased patients. And the last example before we move on to the final research area. And in order to illustrate the value of neuronal activity module, I wanted to show you a model we developed to study drug-induced attenuation of network connectivity. In this example, we studied the effects of the chemotherapeutic drug Taxol, which is given intravenously to cancer patients. Unfortunately, one of its side effects includes neuropathy. We model this by treating echoculture of neurons with astrocytes with increasing concentrations of taxol, from 1 picomolar to 1 micromolar. Here you have two representative videos of the 3-minute epoch of a vehicle and taxol-treated wells. You can see the detrimental effects of the drug at day 10 post-treating. The non-active orange nodes represent dead cells in which calcium has leaked, and as they are not bursting, the software is not segmenting them. As you can see with the plate view visualization, we had 3 replicates per condition, and here we have a summary of the 3-minute epoch of all the treated wells at 4 different time points. The first panel on the left shows the summary calcium trace of all wells at pre-treatment. 14 days post-infections, we observed that network had developed as the number of active nodes had plateaued, and the correlation was at least 80%. We then knew that it was time to treat with taxol. Represented in the next 3 panels, you can see the 3-minute calcium traces at day 5, 10, and 15 post-treatment. The wells we saw videos from before are highlighted in orange. Using this visualization, one can clearly see that the effect of taxol is concentration dependent, as expected, but also time dependent in a chronic way, as you can only start to detect the effects of taxol at low concentrations from day 10 or even 15, illustrating the importance of long-term chronic studies. This effect would have been missed using a short-term drug application. This data, as mentioned before, can be concatenated and represented using time courses and derived concentration curves. The time course on the left shows the change in the number of active objects with time. You can see the network stabilizing for 14 days before treatment was performed. Data was normalized to this pre-treatment value, allocating it as 100%, and deriving from it a percentage change in the number of active objects. It's easy to visualize the concentration and temporal dependent effect. The concentration curves in the middle and the IC50 graph on the right-hand side confirms the temporal dependence of this drug, showing how sensitive the neuronal activity readout is. Now, one can think, is this lack of activity caused by taxol killing the neurons? Well, in order to further elucidate the effect of this drug, we ran a parallel study, this time using Neurolite instead of Reagent, so to look at the neuronal growth and to understand the effects of taxol on morphology rather than on function. The bottom curves show how taxol affects neuronal length in a concentration and temporal manner, but with a slightly different profile as the functional effect, now in the top. The table and its graph show how at the top 3 concentrations of taxol tested, the detrimental effect is fast, within 4 days, and the IC50s for neuronal length and activity compared, meaning that these top concentrations in the nanomolar level are toxic enough to affect both morphology and function. However, as the concentrations decrease, you can see how taxol affects function in a more selective way, and the impact on morphology is much smaller, to the point that by day 10, neuronal activity is affected 20-fold higher than neuronal length, meaning that at these low concentrations, the neurites are still there, but they're just not communicating. This is one example on how by combining different applications, one can obtain greater biological insight. So changing subject, we are now into the last core application I wanted to discuss today. We are now into the neuroimmunology world. This slide shows you a cartoon and the correlated image, IncuCyte image, of an effector cell, in this case, human iPSC macroglia from Essen BioScience, engulfing a target, in this case, proto-label apoptotic neuro2A. We can see how the macroglia senses the presence of the dead neuron, approaches and engulfs it. As we know, the acidic environment of the phagosome derives into an increased fluorescence, which can be then imaged and analyzed over time, enabling the quantification of phagocytosis. The protocol is a simple feed of pre-label particles to the macroglia cultures, with no washing, fixing, or lifting of the cells. In this video, one can see the process in action, and the graph on the right shows a simple validation, indicating that the greater the number of proto-label apoptotic neurons one feeds to this macroglia, the greater the orange signal, symbolizing more phagocytosis. So changing slightly the type of food one can feed the macroglia. In this case, and to test validity of the system, we aggregated A-beta peptide and proto-labeled it. We then fed it to different types of cells, BB2 mouse macroglia, T90AG human glioblastoma, or undifferentiated SHs, human neuroblastoma. As you can see on the first graph, and as expected, only the macroglial cell type phagocytoses this peptide. We then tested how aggregation time affected the uptake of A-beta by BB2 macroglia, and we found that the longer the peptide was left aggregating, up to 4 days, the higher the signal was. We then moved on to more precious iPSC-derived macroglia from actual bioscience, and we observed a concentration response in the uptake of A-beta by these effector cells. We tested the uptake of other disease-related peptides by iPSC-derived macroglia to exemplify not only Alzheimer's disease with A-beta, but also Parkinson's disease with alpha-synuclein, or MS using myelin basic protein. In all these cases, there was macroglia uptake in a concentration-dependent manner, however, and interestingly, with different uptake profiles. Myelin was taken up much faster than A-beta, or alpha-synuclein, and even 3 times higher concentrations of alpha-synuclein still yield only half the phagocytosis signal compared to the other 2 peptides. The last slides exemplify, in my view, how these macroglial models can be used to study the neurodegeneration. A last example on this front is how one can use live cell analysis to gain insight on a particular mechanism by using differential pharmacology as a tool. Here we can see a proposed mechanism of phagocytosis of cells published in the Journal of Cell Science by researchers from Cambridge University. We wanted to discriminate between the process of uptake of dead cells, efferocytosis, versus the uptake of smaller bioparticles, and understand whether integrins were important in one or both mechanisms. For this purpose, we fed BB2s with either Frodo-labeled Neuro2As or Inculyte particles, respectively, and co-applied an integrin inhibitor, silengitide, or a polymerization inhibitor, CytoD. CytoD was expected to inhibit both the uptake of dead cells and bioparticles, as it stops any movement from the macroglia. And this is what we see here, that CytoD inhibited both effero and phagocytosis of bioparticles in a concentration manner. Silengitide, the integrin inhibitor we were testing, only blocked the efferocytosis of cells with no effect on the phagocytosis of bioparticles. You can see, however, that the effect was only partial, suggesting that more mechanisms are involved in this process. And to finish the 4th research area, I'd like to exemplify the use of our chemotaxis assays to study the ability of macroglia to migrate towards a stable chemoattractant. This assay uses the Clearview 96-well plates, composed of a bottom reservoir and a top insert containing a membrane with 96 evenly spaced pores, some depicted in blue. The assay workflow is quite simple. Firstly, one coats the top membrane with an ECM to enable cell movement and seed the cells. Treatments can be added at this stage. Then the chemoattractant is added on the bottom reservoir plate at different concentrations, and the insert containing the cells is placed on top. And lastly, the plate is imaged and analyzed to gather information on how the macroglia cells migrate through the pores located within the membrane. The IncuCyte takes an image from the top and the bottom of the membrane and quantifies confluence, enabling the study of, for instance, BB2 macroglia migration towards a C5a chemoattractant. And finally, as a look to the future, I wanted to touch on a promising area that we are currently looking at, the synaptic pruning of neurons by macroglia. These videos form part of a collaboration with Senso Biotechnologies. They show the great motility of the human iPSC-derived macroglia and how actively they interact with the neurites from primary cortex. An application that one can automatically see is, for example, studies on plasticity, or the differential effects of macroglia pruning in disease and healthy individuals. Let's finish this talk by imaging how much biological insight one could gain from combining this pruning with the previously seen applications. All right, so as a summary of this webinar and to wrap up, we've seen how live cell analysis is ideally suited to develop and validate cell models to answer neuroscience questions. I've covered 4 general main areas of interest: neuronal cell health, dynamics, activity, and macroglia function. We've seen examples modeling neurotoxicity, neurodegeneration, and neuroinflammation, and we've also briefly touched on how one can gain greater biological insight by combining different applications and pharmacological modulation. Thank you very much for your attention. I really hope that you found this webinar useful. You can find further information in the links on your screen, and you will be receiving a follow-up email with copies of this webinar. Also, we can discuss your thoughts during the question session to follow.